Hello, everybody. My name is Tim Nattkemper. I'm a computer scientist professor at the Bielefeld University in Germany, and I have now the pleasure to tell you a little bit about our citizen science tool called Biigle.Party. First of all, why this strange and a little bit weird name, Biigle? The background here is that in my research, I'm developing new computational tools, AI-based tools, to analyze image and video data, huge amounts of video and image data from remote sensing with a strong focus on marine image and marine video. And part of our research is that we have developed an online tool called Biigle, where that is used by around 4,000 marine biologists, that has been used by around 4,000 marine biologists worldwide to annotate and evaluate their images. And in this project, we had the chance to build a small Biigle version with a very reduced functionality and plus some new extra features that could be used by citizen scientists. And this is what I want to tell you about Biigle.Party. So this is the normal screen of Biigle.Party that is a citizen scientist can see when he or she joined a project. On the right, you see different kinds of biological categories, species, like particular kinds of fishes the marine biologists were interested in when they gave us the data. And the big picture shows one image where the citizen scientist can annotate the different categories of fishes by drawing circles. Next slide. So our basic idea in developing Biigle.Party, was possible to get a domain called Biigle.Party, and so we played around with the name. It's the place to participate in marine biology. So that was the idea. So of course the tool was very important that the tool can be used intuitively, and the instructions are easy, and that citizen scientists see a progress bar, so they get some kind of positive impression and feedback that they're moving forward and coming close to an end of their individual project. There is a small gamification aspect in this tool because people need to have some kind of positive feedback to keep their motivation high. And it's anonymous, so it can be used by school classes, for instance, and that it's easy deployable by the marine biologists to advertise their individual project. Next slide, please. So this is just a short video showing what's happening when you use Biigle as a citizen scientist. You have identified 2 lobsters in these wonderful photos from the Aegean, and the 2 lobsters have been annotated, and you see these green bubbles saying, okay, you get 10 extra points for adding 2 new annotations. And here there is also a sea star, which is now also annotated after switching the categories on the right. On the bottom right, you see the 2 blue and red dots showing your progress in different categories, describing your quantity level and your quality level of annotation. How the quantity is easy to measure. The quality is a little bit more difficult, but I do not want to go into algorithmic details here. Maybe we have time to explain this in the breakout session. Next slide, please. So we had 4 projects. One project was performed by 67 citizen scientists. The 3 other projects were performed were joined by a lower number of citizen scientists. Nevertheless, we were able to collect a huge amount of annotations in these images with thousands of circles drawn by citizen scientists. But then you have to ask the question to yourself. Thank you very much. For activating this, that how good was the performance of the citizen scientist? So did the citizen scientist find and mark everything an expert would find? So if there are 10 fishes, did the citizen scientist find all the 10 fishes? So this is the recall. If they only have found 8 of the 10 fishes, then the recall is 80%. The other question is, did the citizen scientist mark anything wrong? So if there are 10 fishes, nevertheless, the citizen scientists have made 15 marks in the image, then the precision is only 66%. To illustrate this a little bit more, Next slide, please. Thank you very much. So a citizen scientist has drawn 4 annotations here. Next slide. There are 3 true positives here. These 3 fishes are correct. Then there are 2 false negatives. So 2 fishes were not annotated by the citizen scientist. It was overseen. And 1 false positive. So the user has made an annotation which was wrong. So next slide, please. And based on this, you can compute the recall, next one, and the precision. So the recall is how much did I find or how much did I detect? And precision is how clean are my results? So next slide, please. Next slide. So this is on the left, you see an image with all the annotations of all the 67 citizen scientists who have evaluated this image from the EACEA observatory and close to the Catalonian coast. And you see here that there are lots of circles around fishes. There are also huge circles, which were false positives, of course, some mistakes done by due to interaction. And on the right, you see the recall and the precision for the individual 4 classes, the 3 different species of fishes and the 4th category, unknown fish. And you see that the recall is very good, but the precision could be a little bit better. But this will improve when more users join the project and you can analyze and you can fuse more results. Right now, we only count those annotations by citizen scientists that have been confirmed by at least 3 citizen scientists. So if there's only 1 circle drawn, like you see here in this image on the left, we don't count it. Below, you see the recall and the precision along all the images in this experiment. So you see the recall is always quite good. The precision is not so good. And the precision is jumping up and down, up and down. The reason for this is that the turbidity in the water is changing. So the expert, because these results are compared to an expert annotation, is sometimes not counting the small fishes in the background, and the citizen scientists try to find as much as possible, so they count those too. However, this could be filtered out later quite easily. So this is basically a quite good result, I would say. So on the next slide, you see now a much more difficult image domain from the Svalbard experiment. So this is a downward looking camera on the sea ground, on the bentos. And there you need more, there the visual experience of the normal citizen scientist is much smaller than, of course, annotating fishes in the OPSI images. Here the people have to learn a very difficult task of annotating branching fauna, sponges, sea squirts, and soft corals. And they were provided with some examples, as you have seen in the small video. And we have a second challenge, that only a small number of citizen scientists, around 30, have joined this project yet. So this is the reason why if we apply our threshold of at least 3 citizen scientists need to agree on one thing, that our recall is going down and the precision is more or less okay. Here in this data, you see strong changes up and down, up and down, up and down. And in image number 14, unfortunately, we did not have any expert label to compare. So this is the reason why in the upper figure, the precision boxes are quite low. Actually, they would be much, much higher if you would have annotations for this image. So this image number 14 spoils a little bit on the results. So the upper figure with the boxes is a little bit misleading. Usually in reality, the results were a little bit better. So here we learned a lot about the task complexity and the task difficulty in citizen science and how you need to adapt your training material to that. Nevertheless, at the end, it's a good thing that we have learned something and that we have these numbers and that the people were happy to use our tool. So next slide, please. All of our experiences, or most of our experiences and findings we have collected in this wonderful handbook. You can download. I guess there's a QR code on the next slide, where we have tried to compile all our ideas and all our experiences in this project. So, and that's it for me right now, and I hand over to Caroline again.
Thanks so much, Tim. And yeah, we just put the link to the handbook in the chat as well. And now I will say to Autun, you're up.
Hello, everybody. Can you hear me? Yay, I have microphone control. Brilliant. Okay. Yep. So I'm a marine image person. I suppose that sometimes I'm one of these experts that Tim was talking about. I made that bad Norwegian data set that we just saw, but I'll talk about my experiences with image analysis and citizen science now. So can I have the first slide, please? Is it worth it? Well, everyone's busy, and to plan a good citizen science project is also slightly time consuming. So it really is a question about whether you actually want to use a citizen science based approach to your work. And I'll talk about that now. Okay, so what I do is I tow a very expensive, heavy camera above the seafloor. That's the one on the left there. And what that does is it takes a photograph every 20 seconds throughout the deployment, and sometimes we deploy this thing for more than a day, so we get many, many photographs of the quality on the right. So this is one from Antarctica, a very busy bit of seafloor, lots of in this case tunicates. So you can see there's a lot of things going on in there, and of course that's too much for a scientist to analyze. Okay, next slide, please. Oops, someone's microphone's going crazy. Okay. Someone got a kid? That's not mine.
I can't see whose mic that is.
Okay, it's gone now.
Okay.
Okay, so is it worth it? So I see citizen science as two things, really. It's a way to analyze lots of data, but it's also a great way to get people involved in what might be a newsworthy story that your institute is studying. Now, we're the Alfred Wegener Institute, which is a German government-funded institute, and we should take into account things that are interesting to the population in general. And citizen science projects, you can demonstrate it if people are interested in taking part in your work, like here. So 2 days ago, on our news page, this is a screenshot from our news page, is the report on a citizen science project. This is on our main institute page, where citizens have collected litter from across all of the German beaches. Now, that's impossible for even a large group of scientists to do. So this is a good example of some work which could not be done without citizen participation. And what they did is they collected a certain amount of sand from certain beaches and sent that off for analysis. It's a great, focused, straightforward piece of work which is repeatable. Next slide, please. So with that awful image set that Tim demonstrated a minute ago, where the levels of data were really bad, the circles all over the place, that was because we tried to ask the people too many questions, perhaps. If you were simply saying, is there a fish in this photograph, the citizens would have been able to label much better, because it's an easier question. So if we could get the next slide, please. This is how an expert sees one of those slides like Tim. So they've gone through and labeled every animal on there. And that's a huge amount of work for a dedicated student whose PhD relies on them doing it. So it's not something for a citizen to do. It's problematic to learn all those animals. To be honest, it's really boring to do this labeling, and it's not answering a clear bit of research. As you label all these animals, in your mind, you're not going to be able to remember when you look at a trawled and untrawled area of seafloor whether you've got less of the pink animal than you did on the last slide. There's too many variables for you to compute in your mind just by doing the work. If you go to the next slide, please. But if you're counting like this, just fishes, and you've got images labeled as trawled areas of the deep sea seafloor or not trawled areas of the deep sea seafloor, when a citizen scientist or a scientist has looked at maybe 50 images, in their mind they're already thinking, there's much more fish in the untrawled area. Actually, in this case, it might not be the case, because these particular fish are using the old trawl marks that we see in this image as a sort of highway. So where the fishing has happened in the past, there's actually more fish in certain places in the Norwegian margin, which is interesting in itself. And being as how I've got 140,000 images like this from my robot submarine that I sent down for this work, it's impossible to expect a human to do that. Okay, next slide, please. Okay, so right now I'm planning a citizen science project. So if you go to the next slide, I'll tell you about it. So as some of you may have known, we discovered with this camera these nesting ice fish in Antarctica. And these are 60 centimeter big fish that sit on their eggs and guard them. And we're going to go back and map some more of this. If you get the next slide, please. So we did a paper about the discovery. In the past, they only found 60 of these nests. Now we know there's 60 million in this particular area, which is really great. Next slide. And this is how they look. You can see them all across the seafloor. And you can see it from this image about 4 meters above that there's some blue eggs underneath. And I'll show you a quick bit of video now, maybe, if that works. So you can see all the different the fish guarding their eggs. And what we thought might be a nice idea and a straightforward idea is to get citizens to label these images taken from these videos, of where the fish are and whether they're guarding eggs or not, or if there's a dead fish. That white one there is a dead one, for example. It's tired itself out guarding the eggs and then it's died. So it's a straightforward question. And the reason that's interesting is because we're going to go to the southern extent of this nest. If we go to the next slide, please. Yeah, so here we see this green area, and that's where the fish's nesting is going on. And we found the fish nest near the top of that green area. And now we're going to look at the bottom of the green area, and we'll get the citizens to label just 4 categories: ice fish, dead ice fish, nest with eggs, nest with no eggs. Very straightforward. And if you go to the next slide, this will really be good at helping us out, because it will allow us to do many images and get a large number of analyses. And I think that we'll have good precision because it's quite a simple question we're asking the participants. And on top of that, we'll also get involvement as we do our news presentations, because we're thinking this is going to have a lot of outreach and a lot of interest in the public. So we will allow the citizens to link directly from our news page to Biigle.Party. Ideally, we haven't asked Tim yet to label these fish directly. So when you read an article, think, oh, that's very interesting, they can click through and do a few labels, just even a few fish photographs, and then they've participated in it and they feel like they're contributing to it, which I think is a nice thing, and will allow us to go through all the images I'm going to collect at the start of next year very quickly. Next one, please. Advice for researchers thinking about using CS for their research. That's what I was asked to mention. If we go to the next one, please. If possible, think about using citizen scientists before you start collecting data. You don't want to be taking a huge area with your images, for example, or you want to make a certain small image, cut the images into small bits so citizens can look at them easily. You want to make sure the lighting is equal so people can label things easily and learn what things look like. So try and think about how it might be for you, people who are not experts, to analyze your data before you start the work. And the next one. I think it's good to, if you can, to make your research question current and interesting. Now, this could be to a target audience of citizen science candidates, for example, divers who go and visit a certain area, like we have in some of our projects in Into the Deep. But it could also be broader, like lots of interest now in biodiversity, global warming, plastics, as we've seen. So if you make it current, you can get more involvement and a bigger number of people may want to work on your data. Yeah, next one, please. Try to make it so make the work so that the citizen scientists can see that what they're doing is useful right from the start. Just when they start labeling a few images, they start to think themselves, oh, this is different to the last image. Or when the light, like we saw in that one earlier of the Barcelona fish, maybe when the sky is darker, you get a different sort of fish than when the sky is lighter. These are things that the citizens can think about while they're doing the work, which I think is a nice thing. So I try to do that with my work as well, I think. Next slide, please. Oh yeah, it must be fun, or it should be fun to do this. It certainly shouldn't be a hassle. No one wants a bad interface to work with in their free time. It should be silky smooth. And that's what I like about the Biigle.Party system interface, is that you can make it very easy what people are doing and straightforward. And I think that's really a good thing if you're going to take people's time. Okay, and the next slide, please. Yeah, so that's really all from me. Like Tim said, I've made some of these suggestions in the same booklet that Tim mentioned. It's great for me to think about the images I take. Like here we see here at this deepest part of the Arctic Ocean, and we sometimes see bits of detritus, be that dead sponges or algae that's flown down from shallow water. And there's always a question that citizens might like to answer, like, these interesting anemones are clearly on this rubbish, and then you can see these little sea pigs are crawling closer. These are sort of things that might be nice as a small citizen science project and lead into a master's project or a bachelor's science project. So there's lots of interesting questions that can lead into real scientific publications that you can think about with image analysis. And that's it. Thank you.